It's 7:02. I think we can begin. Hi, everybody. My name is Marion Snively. I'm a wildlife educator for the Alaska Department of Fish and Game. I'll be hosting tonight's presentation. And Burl Dickman also is a wildlife educator, and she's going to co-host. I want to introduce our presenter, Dan Thompson. He's a wildlife biologist with the Alaska Department of Fish and Game at the Kenai Moose Research Center. Dan has a PhD on moose thermoregulation from Texas A&M University. He has an MS in caribou and reindeer nutrition from the University of Alaska Fairbanks and a BS in wildlife biology from Colorado State University. He currently conducts research on both captive moose at the Research Center and on wild moose on the Kenai Peninsula, integrating nutrition, physiology, and behavior into moose research. Please, for some housekeeping recommendations, please keep your camera microphones off during the presentation. Dan will answer questions at the end of the presentation, but feel free to write them in the chat anytime during the presentation, and they'll be answered at the end of the presentation. Okay, I'm going to turn it over to Dan.
All right, thank you, Marianne. Again, thank you for having me here today. My name is Dan Thompson. I'm a wildlife biologist at the Kenai Moose Research Center. I was asked to give you a quick history of the research center here and some of our current research and how that is being applied to wild moose on the Kenai. Let's get my slides to work here. Here we go. So for those of you that are not familiar with Alaska, we're located on the Kenai Peninsula in South Central Alaska. On this map, you can see our research center is located here in the star, and it's in the northern part of the state, or part of the Kenai. We do also do research in game management. It's units 15A, B, and C, and we'll be talking about a little bit of that towards the end. So some history for the research center. Shortly after Alaska became a state, the land management agencies came together, those being all the federal agencies, the National Park Service, Fish and Wildlife Service, Bureau of Land Management, as well as the military bases, along with the state of Alaska. And they had these interagency moose meetings. And out of those meetings, they decided that we needed to learn more about moose before that they could actually manage them. And they decided to establish a captive facility on, at the time, was the Kenai National Moose Range. And the main thing that they wanted from that facility was to gain knowledge of the interrelationships of moose in their environment. So the facility began construction in 1966. They started putting in the fence lines, and over the next 3 years, they were able to fence in 4 square miles of fence that was completed in August 1969. Now the facility, we started doing research in 1968 when we had about 3 of our enclosures finished with the fence line. And at that time, through 1980, it was a joint research operation. The Alaska Department of Fish and Game was in charge of all the animals and animal research, whereas the Fish and Wildlife Service, and it was actually the Denver Wildlife Research Center, was in charge of doing habitat research. And so the early facility, they basically had to trap wild moose along the fence lines, and they would manipulate densities between the pens. Here you can see a biologist that he's darting the moose in that fence line trap. They would then go in and take samples from them, put on identification tags, and then different ways that they used to weigh them back in the day. So here you can see the sample's got an ear tag with an identification tag. This one's got a neck collar. So this is the way that they did some of the research at the beginning. And a lot of it focused in on starting to understand some of the different things about moose that we don't know a whole lot on. So developing and testing new techniques, moose productivity and physiology, moose calf mortality study. But the one that's stuck with the research center since the inception is developing and testing new techniques for moose management. And that's, we still do that today. Some of the early research from the Fish and Wildlife Service, again, it focused more on vegetation. So looking at nutrition of cranberry, looking at browse production. And so this research went on until about 1980 as a cooperative effort. In 1980, with Alaska Native Interest Lands Claim Act, the Kenai National Moose Range was then converted to the Kenai National Wildlife Refuge. At that time, the operation and the research side of the facility largely came to the Alaska Department of Fish and Game, whereas then the Kenai National Refuge was more for facility maintenance. So since 1980 to present, the Alaska Department of Fish and Game has been running the facility and conducting all the research out there. And so there's been a wide variety of research since that time. In the 1980s, a lot of it was focused on energetics and nutrition. You can see here some examples for energy requirements for moose and metabolism, as well as looking at digestibilities of different browse species, digestibility of protein, looking at formulating diets for these animals when they're having them in captivity. And another thing that really came out of the 1980s was what they call the animal indicator concept. Al Franzman was one of the first directors of the facility, and he coined this term the animal indicator concept is based on the premise that animals in a population act as indicators of the status of the relationship with their environment. Now, this idea is that you can go up to an animal and take a physiological or morphometric measurement, and then if you go back to that animal again, down a month later, a year later, you can start tracking changes in that animal which are related to its environment. And so they started really looking into this in the 1980s, looking at body condition of animals. How can we measure them? They looked at using doubly labeled water. They did bioelectrical impedance. They also looked at morphometric measurements, so measuring chest girth or length of legs related to body size. But one of the methods that they really keyed in on was this using an ultrasound to measure rump fat measurements. And so this was a technique that was pulled from the livestock industry, measuring fat, and we were applying it to moose at this point. And the one that study that Tom Stevenson did at the time was using that ultrasound to determine how much fat this animal has, and then validating that for total body fat. Now, this technique we still use today, and it has been applied to moose in the lower 48 and also has been adapted to most of their ungulates in the lower 48, as well as Alaska, so including caribou, bighorn sheep, and some of your other deer species in the lower 48, as well as elk. So this method is when these animals are sedated, you can go in and take this ultrasound and you can measure the depth and the thickness of that fat on their rump, and that's an indication of their nutritional condition. And so this is a method, again, that was developed in the 90s, and we still use it today. Some other things that we developed here and worked on in the 90s and early 2000s, Chris Huttermark was really leading the way in genetics and looking at diversity and evolution in these different moose populations. We also came up with different ways to determine pregnancy from blood samples or fecal samples from these animals. And then also looked at different management implications. In the 1990s, Alaska passed the intensive management law. And so the staff here looked at things such as the considerations for that, as well as selective bull harvest. And a lot of that early research has been put together into this large book, The Ecology and Management of North American Moose. This book is over 700 pages, and it has a lot of stuff that was done at the research center, including all the things we just discussed. And just to point out, those two editors of this book, Al Franzman and Chuck Schwartz, they both were directors of our facility. Al was the first director and Chuck was the second director. So a lot of our research that we did in the early days went into this large book. So to give you, now we're kind of up to current day. Our facility is staffed by 3 Fish and Game employees. John Kraus is the physiologist on site. He's the director of the facility. Jason Rupp is the Fish and Wildlife Tech 4. He lives on site. He takes care of the facility, make sure everything's running smoothly. And then myself, Dan, I'm the wildlife biologist. I coordinate most of the research that we have going on at our facility. So our facility is unique, and in the last 20 years or so, we've really focused in on hand raising our moose, and this allows them to become tractable animals when they become older. So this is a lot of work. These animals, we get them 1 day old and we raise them up through weaning. We train them to do things like get on scales, being used to us being with them in the field, sitting next to them, walking with them, so that we can do research on them once they grow up and become adults. And some examples of that is this here. Where this cow just gave birth to this calf, and we're able to stand next to it and weigh that calf and return it to her. This top figure, we took the GPS collar off that moose, and the grad student's downloading the data while she's sitting and waiting, collecting insect data or observing these animals for what they're eating up close. And so this is just a video of one of our moose shiner. We put a GoPro up on top. I'm not sure if it's going to play, but you can see we've trained her to come up to this weigh scale for a little treat of pellets. And this allows us to walk her up onto this scale and get a body mass on her. And it's something that you wouldn't normally be able to do if these animals didn't have all that effort put into them as calves. And so again, we invest a lot of time when these animals are young to be able to get data like this, because this is very hard to get something like that. So just to get into some of the recent research, I'm going to focus mostly on some of the stuff we've done on thermoregulation. And again, this goes back to that animal indicator concept, that a metric of the animal is an indicator of its relationship with its environment. In this case, we're looking at their body temperature as a metric with their relationship to the thermal environment. There's been a lot of interest in the lower 48 in regards to how warm temperatures are influencing moose, but we felt like we needed to back up and find something that we can actually track with the animal. And so we wanted to look at, we needed to establish what are the baseline body temperatures and figure out how they can get rid of body heat, either physiologically or behaviorally. And so first thing to do that, we needed a way to measure that body temperature. And so we validated 2 different methods here at our facility. First, we used a vaginal implant transmitter. These are used predominantly, you put them in a pregnant female and it's expelled at birth, and that gives the researcher an indication that those animals gave birth, and we can go and see how many calves they have. So in this case, we added in a temperature logger so we can get body temperature. The second one is a mortality implant transmitter. This is designed to reside in the reticulum of the moose, and this is designed as a mortality implant. So some places they want to know exactly when an animal dies, and so this records the activity within that animal, and if it stops moving, it sends a signal to the collar and transmits that. We're able to have a temperature sensor in that as well. So we're adapting some of the technologies that are being used for other methods to collecting body temperature. And with that, we wanted to first establish what are these baseline temperatures in moose. Most of this had been collected by moose that were immobilized and one-time measurements, not collected over time. So what we were able to find, this graph, you can see this is the course over 1 year, and this is the body temperature of these animals. You can see here in the wintertime, their body temperature is lower, and their body temperature increases in the summer and comes back down. So we see a seasonal variation in body temperature. And this was something that had not been documented in moose yet. We also see a daily change in body temperature. So over the course of a day, their body temperature will vary. And you can see on this graph, over a 24-hour period, it'll vary over the course of that time frame. It also changes by season. So in the summertime, that amplitude that changes is greater than in the wintertime. So these are things that we need to know as baseline measurements before we could start determining when these moose might be heat stressed, looking at their body temperature values. So then next, how do they regulate this body temperature? For this, we really looked at physiological responses. And I'm going to go over 4 of them with you today: heart rate, respiration rate, exhaled air temperature, and skin temperature. So first, for heart rate, some of our moose, we were able to put this polar heart rate belt on. This is designed for horses, so we could follow them around and get heart rate as they went about their day. Other times, we'd have to follow them around until they laid down. And when they're laying down, you can actually see the pulse in this vein in their neck, so we could count it manually. So some of the results from that, you can see this is heart rate increasing here with warmer air temperatures. And we found a negative relationship. So that means that as air temperature increases, their heart rate is actually decreasing. Now that might be counterintuitive to you, but there's a few explanations for that. One, during warmer temperatures, they're probably drinking more water, so they're more hydrated. Therefore, their blood volume is greater. So it's going to slow down their heart rate with that, pushing that more blood through their system. The other one is that when your heart is beating really quickly, every time it contracts, it's pushing out a smaller volume of blood. And so if you slow that heart rate down and allow that ventricle to fully refill before it contracts, you're pushing more warm blood out to your extremities in a way to get rid of that body temperature, that body heat to your using your skin and other methods like the respiratory tract, which I'll show you in a minute. The other thing we see is time of day. Over the course of the day, their heart rate is the lowest during midday. And this goes back to this body temperature. When we look at the 24-hour period of moose body temperature, we see that daily fluctuation in body temperature, and it's the lowest during the middle of the day. So they're doing this physiological mechanism to help bring their body temperature down. When we look at respiration rate and exhaled air temperature, respiration rate, you can count. You can see here's just an example. You can count the breaths. You can see, you can also do this without the FLIR camera by counting their hip flank movements. For the air temperature, every time they exhale, you'd follow the moose around. And since they're so used to us sitting next to them after they lay down, you can take this fine wire thermocouple and I could stick it up in their nostril and I could measure the temperature of the air coming out every time they exhaled. So for respiration rate, what we see similar when the air temperature increases here, the respiration rate is increasing. So that means when it becomes warmer, they're breathing faster. Now we also see this time of day effect as well. So during the middle of the day is when they're breathing the hardest. And again, that comes back to this time of day, trying to bring that body temperature down. If we look at the exhaled air temperature, we'll talk about this side first. This is vapor pressure. That's also another term for humidity. So higher humidity, it decreases the amount of heat that they can lose every time they exhale. This has to do with the amount of moisture in the air. So the way that you get rid of heat from your respiratory tract is you're evaporating water off the surface, and that method of it, when you evaporate it, it takes that heat with it. So that water needs to go into the air. If the air is very humid, they have a harder time doing that. And so that's why you see this decrease in heat loss with more humid conditions. Again, we see time of day. They're losing more heat from exhalation during the middle of the day, again, pushing this temperature down midday for their body temperature. The last one I'll show you is the ear temperature, the skin temperature. So during the summer, their ears are largely void of any hair. It's one of the first things that they molt in May, and usually doesn't have much hair on it until end of July. And so we were able to go out and take pictures of this, and you can see this big vein right here is bringing the cold blood back into the ear. And this is an artery bringing in the warm blood. You can see that in this picture. So the brighter color is the warm areas. That's bringing the warm blood in. And this is the vein taking it out. So we were looking at the difference in that temperature of the vein or the artery compared to this whole area. And the idea is the closer those two values are together means that they're pushing more warm blood through vasodilation. So they're pushing it into their skin and warming that up for trying to get rid of it. And this, we've seen a similar type thing. This, again, if this value is closer together, that means that the whole ear surface is getting closer to that artery surface. And we see that again, time of day effect. Again, as it's something these animals are trying to do to bring that body temperature down during midday. And that's more than likely because they are anticipating those warmer hours in the afternoon, particularly here in Alaska, are kind of long days. They get fairly warm in the afternoons and early evenings. So that was just a very quick explanation of some of the ways that these guys can regulate body temperature through physiological mechanisms. Next, I'll just go through some behavior mechanisms that we can see what they're doing and how it influences their body temperature. So for this, we use the rumen boluses. So that not only gave us a body temperature, but it also, you could see when they are taking a drink, you'd see a spike that would go down in their body temperature and their rumen temperature, so we could document drinking events. These animals also had a GPS collar on them. That gives us habitat use as well as movement rates. So how fast are they moving in these different habitats, and how does that influence their body temperature? So this graph, there's quite a bit going on here, but I'd just like you to focus in on this hour of the day we're going to be talking about from midnight to 6 in the morning. This one here is the change in rumen temperature. So anything below this dotted line means their rumen temperature is decreasing. Anything above it means their rumen temperature is increasing. During those early morning hours between 0 and 6 a.m., their rumen temperature is largely decreasing. However, we do see that their movement rates are increasing at that time. And so that's kind of counterintuitive. Usually you think if you're moving, you're generating heat. How is their rumen temperature coming down? We don't see a lot of drinking events during that same time period. So they're doing something else that is causing their rumen temperature to come down while out actively moving on the landscape. So it comes down, part of it comes down to the habitat they're in. And this is a very large graph. I just want you to focus in on this one. This is the early ceroboreal forest. You can see it's the only graph where this rumen temperature goes below compared to all these other ones with movement rates. And we'll zoom in on that. So again, this is the movement rate. So how fast are you moving? And this is the change in rumen temperature. When you're not moving, when they're bedded, you can see their rumen temperatures are increasing. And that's more than likely because they're laying there. They have diet-induced thermogenesis, and it's bringing up their body temperature. But when they have these low to moderate movement rates, we see their rumen temperatures are decreasing. And once you exceed about 600 meters in an hour, then you can see that influence of movement increasing their body temperature. In this early ceroboreal forest, that's basically areas that have either been burned or crushed. It's basically their grocery store. So they're out there eating fireweed, aspen, a lot of your other browse. Another component to this is when they're doing it. And this is the probability of being in an early cereal habitat over the course of the day. Again, here's that early morning period. They're out there in this early cereal forest quite a bit compared to all of our other habitats. So what's going on is they're out there eating, and if they're eating at that cool part of the night, they're ingesting a lot of cool and usually wet vegetation from the dew on it, and they're getting this large influx of cool, wet vegetation into their rumen. And that's why you're seeing their rumen temperatures decreasing while they're actively out foraging. So that's one way that we can see that a behavior of foraging during that time period is influencing their body temperature. So that's a quick overview on some of the thermoregulation research that we've done. From all this research, it was really a jumping board for some other research that collaborators wanted to come in and do with us based off of what we've done. And so most of that, we have 3 studies that are still using this body temperature currently. 2 of them are looking at pairing body temperature with hormones during the rut. So some of our collaborators want to know if we can see peaks in body temperature during rut in these females to know when they're going into estrus, and then tying that with some of these hormones like fecal progesterone or urinary luteinizing hormone to identify those as when the animal's in estrus. We also have collaborators that are interested in heat loss in antlers in the bulls. And so I'll give you a couple of examples of these studies here. So the first one was Jennifer Hoy Peterson. She's a master's student out of Norway. She was interested in looking at the peaks in body temperature associated with estrus and tying that to fecal progesterone. In Norway, they have a lot of moose that they have these temperature sensors in, and they wanted to get an idea when these animals were being bred, but they didn't have a good way to do that because those were wild animals. So they came and did this project to try and do that with fecal progesterone and body temperature. Jennifer was able to start to key in on some of those peaks in body temperature, but the fecal progesterone wasn't as fine a scale as they needed to really identify those estrus peaks. So Lucy Lemire is another PhD student out of Norway, same place. They decided they wanted a second run at this, but looking at a more precise hormone, urinary luteinizing hormone. And so that's something where we go out and we collect urine from these animals, and then we hopefully will be able to get a better idea when these animals are in estrus, and then tie it to their body temperature from those sensors. The last study on body temperature we have going is with Rebecca Levine out of the University of Wyoming. And they're interested in how bull moose can lose heat through their antlers. They did some work with GPS moose in Wyoming that showed that bulls chose different habitats than cows during the summer, and those habitats a lot of times were more windy than the ones that the cows chose. And one of the theories is that those areas with a higher wind are helping them get rid of heat, particularly off of those antlers. And so Rebecca came up and she used FLIR cameras to look and document the amount of heat coming off these guys, and then did some 3D imaging of the antlers to try and get a surface area and quantify that loss. Some other work that we've done, this is Bridget Benedict. She was a PhD student. She just finished up. She's looking at the effects of biting insects on moose. And so for this project, Bridget went out and trapped insects in all of our habitat types over 2 summers and was able to kind of map which habitats have certain type of insects that harass these moose. She also went out with a sweep net and followed these moose around and collected insects directly from them. And she also looked at these sores. So here, at least in South Central Alaska, you can see all the flies on this backside of this moose. These moose flies, horse flies, deer flies, they create these open sores that are on the moose pretty much from first part of June through end of August. And so Bridget also quantified some of the issues that can arise from that, including there's some microflare and legworms that are transmitted through these sores. Some other stuff that we've been working on are validating biologgers. So any of you that wear a Fitbit, that's a biologger. It's any type of miniature device that records acceleration or heart rate or magnetometer. So for in these moose, almost all GPS collars now come with some form of an accelerometer in them. We also put magnetometers. Magnetometer tells you which direction the animal is going. And then some of our animals, we were able to put subcutaneous heart rate loggers in so we could get heart rate over the entire year. So for the first use of accelerometers, Jane Denninger out of Mississippi State came up and followed our moose around for 2 months, and I was working on validating behavior based off of these accelerometers. So these accelerometers are triaxial, meaning that they record motion front, back, side to side, and up and down. And Jane would follow these animals around and document the behaviors they're doing, and then she can tie the accelerometer values to those behaviors. So that way, at some point, we should be able to put this on a wild moose and infer what they're doing in those places from the accelerometer data. The second part of her study is looking at magnetometers. And again, this is the direction that the animal is going. And so traditional GPS collars will take a point, let's say every hour, and you don't know what happens between those 2 points. Well, now with the acceleration data and the magnetometer, you can start to determine the path that they went between those 2 points. And so that is some stuff that we're looking at, and hopefully we'll have before too long. Another student that working on biologgers is Teresa Kirchner. She's doing a similar type work with behavior, but a different type of accelerometer, and trying to infer the different behaviors from the accelerometers. But then it's also tied to heart rate and looking at energetics. So if we can determine behaviors from the accelerometers and then tie that to changes in heart rates for each behavior, you can start looking at energy budgets of these animals. Now, because we have things like heart rate loggers in these animals, we try and maximize the amount of data that we can get out of them. So as kind of a side project of this was to look at the influence on drones. So a lot of people know, see the behavioral response that a drone does to an animal, but nobody really has a physiological response. So this animal, Sky, she's got a heart rate logger in her as well as a temperature logger. And there's a drone above her, and you can see her starting to react to that drone. Interesting enough, it's the same reaction that she has to like a horsefly pestering her. But we do start to see these reactions from a drone. Now, I sat with her for 10 minutes prior to the drone coming in, and she was perfectly calm. So we're trying to document those physiological responses and maybe determine minimum heights that you need to be if you're trying to observe animals with drones. And then our last grad student that we have at our facility is Garrett Savory out of the University of Alaska Fairbanks. He's looking at stable isotopes, and basically he is looking at the different values of nitrogen, carbon in the plants that they consume, and then looking at those values in their blood profiles and starting to determine some of the different nutritional pathways that those various nutrients, the nitrogen and the carbon, are going into the animal. So that was a pretty quick overview of the history of the research that we're doing at the Moose Research Center and the current research that we're doing within our facility. We also do research on wild moose on the Kenai Peninsula. And so a lot of it has been in response to intensive management, and then more recently in response to large-scale wildfires on the Kenai. And so what I'm going to quickly talk about and touch on today is, this is a very large figure, but we're keying in on this nutritional condition. And again, that's what we talked about with measuring rump fat, so knowing how fat an animal is, and then looking at how that influences what we call life history components. Those are things like offspring survival, age at first reproduction, pregnancy rates, adult survival. If you add all those up, those influence how that population is either increasing or decreasing. And what we wanted to do was show that the nutritional condition of a population is an indicator of that population growth rate. And I'll also go over some of the other things that influence nutritional condition, particularly the range quality, and explain a few of the things that we're doing with that. So for the nutritional condition, this is the body fat. We talked about this, measuring it with the ultrasound. And this is your lambda. So lambda is your population growth rate. If your population growth rate, or lambda, is greater than 1, that population is increasing. If it's less than 1, the population is decreasing. So what this graph shows you as the amount of body fat in that population increases, lambda also increases. So that's a very direct trade-off, showing that nutrition does influence the population growth rate. Now, this is a fairly new concept. And when I say new, it's because it's very hard to get to this point in the data. This is 11 years worth of data collected in 3 different moose populations to produce this graph. It's very hard to get that long-term data set where you're going back to the same animals and getting growth rates and survival rates and fecundity rates to generate this. And in fact, it's only been shown and published in 2 other ungulate species to date, here in mule deer in California, and then again in bighorn sheep in the same area in California. So we're really excited to see that we're seeing the same type of relationship between our nutritional condition of our moose and the population growth rates. And again, that's just linking these 2 things, nutritional condition to the population growth rate. So now I'll quickly go over a few things that we're doing on range quality. So here on the Kenai, in the last 10 years, we've had some pretty large wildfires, particularly the Funny River Fire in 2014 and the Swan Lake Fire in 2019. And these 2 fires burned a good chunk of the landscape. This one in the Funny River Fire in GMU 15B burned a large portion of the vegetated area in that unit. And then again, we had that fire up here in 15A 5 years later, and it really changed the landscape as far as nutritional condition for moose, the quality of available forage on the landscape. So for this study, again, we were going out and we were catching these animals. This is the same animals each fall. So this is November. This is over time. So starting in 2014 through 2021, this is your body fat. And you can see between 15B, which is the blue diamonds, that's the area that burned in the Funny River Fire, and the black would be 15A, the old Cerro. We don't see a difference in body fats those first 3 years after the Funny River Fire. But here and going forward, we do see elevated rump fats of those animals in the fire footprint. And then here in 2019, we had that fire in 15A, and we see them popping back up here 2 years later. So we are seeing a response in our body conditions to the changing nutritional condition on the landscape. And to take that a little bit further, we have a vegetation map that was put together as a collaborative effort with our federal partners as well as the borough to map the vegetation on the Kenai. And through that, we were able to generate a moose, what I'm calling a moose forage index. It's based off of vegetation cover and based off of vegetation cover of species that moose consume. And so by doing that, we can determine which of these different vegetation polygons are higher quality for moose, and then start analyzing their movements and using of those areas. And so to do that, again, we had the GPS collars deployed on these animals, and this is a home range size for one animal during the summer, so during gestation. And we can take that home range and then overlay that on the vegetation map. And then we can start looking at how much, what is the quality of the habitat within that moose's home range. And then does that influence over the summer, all that area that that moose has went through, does that influence their rump fat that coming fall? And so we did see that as the amount of their home range that had higher moose forage index increased, those animals had higher rump fat. So we're seeing the direct link there from increasing range quality to increasing body condition. The last part is kind of what I call thinking like a moose, so evaluating moose habitat. And so this is, we're going out and we are collecting samples and trying to quantify how much biomass is on the landscape. And then those samples that we collect, we'll be running them for digestible protein and digestible energy. However, we want to key in on just those species that moose are eating. And the thought behind this is, as everybody in Alaska knows, and probably elsewhere, that after a large fire, so this is like a year after a disturbance, and this is the habitat quality. If you have a large fire, the habitat for a moose increases the first 15 to 20 years after that fire. and it starts coming down and declining. Most people, and especially moose hunters, know that, but nobody's actually went out and documented it. So that's what we were trying to do here. So what we do in the summertime, we'll go out 3 times in June, July, and August, and we'll sample the biomass for those species and parts of those plants that moose will be eating during that time. So mostly fireweed and rose, and then you're getting the leaves off of your browse species. And then in the wintertime, we'll go in October and March, and we're mostly doing the twigs of those browse species. However, if there's no snow on the ground at that time, we'll also collect the biomass and nutritional quality for things like lowbush cranberry, wintergreen, and bunchberry. Those are things that they'll be able to eat if they're available and not buried in snow. Now you might be asking, well, how do you know what parts of the plants that these animals are eating? That comes back to our captive animals at the Moose Research Center. You can see this animal is out eating fireweed this time of year, and they're clipping off about the top 20 centimeters of it. You can see as she goes foraging here, she's going to take a bite of a fairly large fireweed right here, and she pulls the whole thing out of the ground, and she clips off. She left that whole bottom stem. So we're only sampling that top 20 centimeters of these plants, similar to what these animals are eating. Same thing for rose. Here's an animal browsing on rose. You can see the parts of the plant that she's eating. We're mimicking that same part of the plant when we're out sampling. Here's another example. Again, this one's eating a fireweed. You can see she raises her mouth up. She's taking off those tops of the fireweeds. But then she's also going to start browsing on those aspen trees, and you'll start seeing how they're stripping those aspen. So again, we're mimicking that same motion when we're collecting our samples, trying to get the same plant parts that these animals are eating during those times. And then this is just another example. This is moose eating dogwood. This is in October before the snow falls, and they're just going through it and picking out all these individual dogwood plants. And so that's the same thing we're trying to do. And so this really gets at trying to quantify these changes in the landscape after these fires. So this is that summer post the Swan Lake fire. You can see it's predominantly fireweed in there. The comparable site next to it that did not burn, it's very closed canopy. The only really forage in there for those animals is this high bush cranberry. And so we're going in and sampling those 2 areas. Same thing for the black spruce sites. Again, largely fireweed here after the fire. Compared to this late cereal stand, there's not very much to eat in there in the wintertime. However, there is quite a bit of cranberries in there that they do eat in the shoulder seasons when there's no snow. So those are some of the things that we're trying to look at and show how range quality influences your nutritional condition, and ultimately tying in those things to influencing their life history components and the growth rates of those populations. So that was a fairly quick overview of everything that we've done here at the Moose Research Center in the last 55 years and what we're currently doing. Again, we started running the facility more or less in 1968. Since that time, we've published 165 peer-reviewed publications, and we've also had 31 graduate students come through our program in that time frame. And with that, if any of you guys have questions, I think I have